Thanks for, well, making this happen. Thanks for the introduction. It's really it is a great pleasure to be part of this community. And this is certainly, at least to me, the most exciting event of the year. I know we're all a little bit Zoom fatigued, but this one is great. And I want to really thank the organizers for keeping the community and spending an incredible amount of time and energy in organizing this. Okay, so I know I'm, preaching to the choir here, and I don't really need to convince this audience that networks are useful, but I still find it instructive to start from the very basics and spend a few minutes on reiterating at least some of the fundaments of network biology, the way I see them at least. And the key metaphor that really summarizes network biology and why it is useful, and that also has inspired much of our own work over the last years, is that molecular networks, they can be really characterized as maps. So these maps, they really guide in trying to understand how biological function emerges from this interplay between many individual components. And in my mind, there are four key ingredients to this metaphor. The first one is that global shape is important. So I'm sure that you recognize Manhattan, this Manhattan Island, just by its shape, and also molecular networks have a global shape. So for instance, this is a beautiful example where this very peculiar circular global shape is related to processes of inflammation and the return to homeostasis. And also more generally, we really learned a lot about how the global shape of network can be related to the processes of how they grew, let's say, over evolutionary timescales. So the first classic example is of course the rich get richer phenomenon that can be related to how gene duplication leads to a certain scale-free structure in these global networks. The second part of this analogy is that location matters. So, the Starbucks here at Times Square sells more coffee than the one on Roosevelt Island. And we also know that the location of a gene within these molecular networks is related to how important it is in a very broad sense. So this initial very seminal work on gene essentiality in yeast protein interaction networks showed that those genes that are really at the center of these molecular networks are more important. And other works have shown the same thing for cancer driver genes, for genes where mutations have a very detrimental effect, causing severe hereditary diseases, genes that are active in early development. So there is a broad body of literature showing that the centrality compared to these more peripheral genes is an important concept in understanding these molecular networks as maps. The third aspect to it is that neighborhoods are important. So, Wall Street is different from Brooklyn. It's frequented by a different type of people, and it serves a different function. And the same is true also for molecular networks, where genes that are related in their function actually aggregate in these local neighborhoods, as shown very beautifully here in this genetic interaction network by the work that Charlie Boone has done over the last 10, 20 years. And the same is actually true also for disease-associated proteins. So they are not scattered randomly or all across this map, but they aggregate in what we would call disease modules. And finally, the fourth part is that distance matters. So it is no coincidence, I think, that the poorest part of Manhattan here in the Bronx is maximally removed from Wall Street. And we also find that in molecular networks, the distance between disease neighborhoods correlates, for example, with shared pathways, but also with similarities in the symptoms that these respective diseases have, all the way to being more showing higher patterns of comorbidity. So all this really is a function of how close these two disease neighborhoods would be on a molecular network. And so in summary, in my mind, these four concepts, they allow us to view molecular networks as maps, and this in turn can be used to address a very broad range of questions in biology and in medicine, which span the entire range from very theoretical to methodological questions all the way to their application. And here are just some examples that, at least in my mind, are particularly interesting. So starting from the very fundamental question on how to even think about causality in the face of these very complex networks where multiple combinations of perturbations play a role in a given disease phenotype, but also in a given healthy phenotype, all the way to how do we think about well, causality across different scales of biological organizations. So how do we move from these molecular networks that we have been studying quite a lot over the last 10, 15, 20 years to networks that would incorporate cells, tissues, organs, and more systemic effects. I think networks also have a say in how we actually think about these very large and high dimensional data sets that biology is now drowning in, all the way to how can we move from a more descriptive network biology to really translate this to very concrete, in our case, medical applications. Right, so for the rest of my talk, I'd like to pitch you some of the projects that we've been running in the last years, most of which are not yet published or hopefully published soon. And I want to start here at this end at the theoretical question of how to think about combinatorial perturbations. And the essential question really is what is the combined impact of a combinatorial perturbation? So I mentioned earlier that this closeness on this molecular network can both relate to either, say, disease comorbidity, so these two representing, I don't know, type 2 diabetes and obesity, but this closeness can also be related to the efficacy of a particular therapeutic intervention, if one of these two would represent the targets of a particular drug. So in one case, really, it's a positive, a desired outcome. In the other case, it's a negative comorbidity. So the broader question is what is really the combined outcome of individual combinations of perturbations on these networks? And this question is really at the heart of many, many concrete problems. So if you think about the interaction between drugs, or if you think about the epistasis of individual SNPs on the genome, or if you think about environmental genetic interactions. So all of these seemingly unrelated questions, in my mind, really boil down to what do we expect the combined final impact of different independent perturbations to be. And my delusion, as Martina said, I have a background in physics, is that there really should be principles out there that would allow us to understand all of these scattered phenomena on the right under one umbrella, which I'd like to think of as some sort of grand unifying arithmetic of perturbations. So over the last years, we have set up a combined experimental and theory framework to actually approach this. So what we do is we use cells as a very simple and well-controlled model system. We then systematically perturb them experimentally by using drugs and combinations thereof, and more recently also by using gene knockouts. And then we use high-dimensional, high-content microscopy to take images of these cells, apply a framework that I'm going to explain in detail, and in the end, by integrating all kinds of available data on these drug-drug interactions that we measure, we hope to close the loop and really learn something on the mechanisms of what causes, how do different individual perturbations converge to a combined phenotype. Okay, so in the interest of time, I selected a few steps of this pipeline that I think are most interesting from a network biology perspective. And the first one is this, where the question is, how do you actually quantify interactions between high-dimensional readouts? So the experimental data that we are getting from these experiments are cell shapes for different drug treatments. So what we do with this is we construct what we call morphological space, in which every possible cell shape corresponds to an individual point in this space. So the axis would be cell shape features like the cell length or the diameter or many others, so that we can then identify a drug-induced change in cell morphology with a vector that would move the cell from one place in the space to another. And in turn, if we have two of these drug-induced perturbations, we can define a 2D hyperplane in this space. So within this 2D hyperplane, we can, using straightforward vector algebra, calculate what is the expected place in this space where a double perturbation is supposed to end up, and then compare it to where it actually ended up. So with this, we can subdivide this very high dimensional space into 18, I think, different regions that correspond to 18 different types of interactions that are displayed here. So they can be, undirected, where you have an entirely different phenotype emerging if two perturbations are present at the same time. But they can also be directed, where one drug would increase or decrease the action of the second, or the other way around. So in total, we have these different patterns. And yeah, I'd like to emphasize that this is really a fundamental generalization of the concepts of synergy and antagonism that are very fundamentally limited to a one-dimensional readout. So if you only know that cells die a little more or a little less than what you would have expected if you have a single parameter that you're measuring, then you can only say, well, they have a positive or a negative interaction. Whereas for these very high dimensional readouts, you can actually have any of these patterns. Okay, so here's the result of applying all this vector-based framework to the imaging data set that we have. So this is really a high resolution. We came up with a silly name, Perturbome, of how drugs interact with each other on the cellular level. So here the nodes in this network represent drugs, and the edges correspond to their interaction. And even though the picture itself on a first glance looks quite messy, it is in fact remarkably sparse. So there could be more than 90,000 edges here. These are all the possible ways in which these some 250 drugs could potentially interact with each other. But in fact, there are less than 2,000 that we determine experimentally. This means that in the vast majority of cases, two drugs actually do exactly what you would expect they would be doing from their individual effects. And even in these rare cases where they do interact with each other, again, in the majority of cases of those, they only interact in one direction. So they rarely engage in crosstalk, but it's almost always one drug modulating the effect of the second. Right. So, and I think, when we first saw that, I was quite puzzled actually that in light of how fantastically interconnected the molecular targets of these drugs are, it is quite surprising to me that the effects of these drugs are very well shielded on this molecular network. Okay, so now that we have this description of how our 300-ish drugs interact with each other, we want to understand it better. So we want to link these descriptive observations on the cellular level to both molecular level knowledge on the drugs and the cells and their targets, but also to knowledge that we have on the clinical side of what side effects do these drugs have, what are known indications of these drugs, and so forth. And the results of trying to link these observed interactions to known biology are on this messy slide that is really supposed to only make you to make two key points. The first one is that on one hand, we can recapitulate a bunch of known mechanisms of drug interaction. So, for example, we recapitulate that some of these drug interactions are likely the result of competition for the same metabolizing enzyme or for a transporter. But we also find a range of unexpected associations, which are summarized here in this second key point, which is supposed to say that these different types of interactions, the positive ones, the negative ones, and these emergent ones, they really have quite distinct molecular fingerprints, meaning that they have different patterns of enrichment across these different biological data that we are using, which suggests that they can be predicted. from orthogonal data sets. And indeed, when we put all this data into a big, reasonably straightforward machine learning random forest classifier, we see that they are actually predictive. It's not great, but it's enough, at least for our mind, to conclude that what we are measuring on a reasonably artificial system of cell morphology and an ex vivo model system has meaning for broader biology. And in my mind, the most interesting part on this is that if we look at what are the actual features that convey the biggest part of this predictability, we find that it is again the features that are related to these interactome-based distances, which are summarized here on the right. So if we have two drugs that really target distinct areas of this molecular network, they mostly do not interact with each other, but they exert their function independently of each other. Now, if they are overlapping, so if the targets really start targeting a related area in this molecular network, this is when they start to also exhibiting interactions, and it is really the precise extent of their overlap that can be related to predict in which direction they would actually interact with each other. So whether it is a positive, negative, or emergent interaction. And yeah, let's say my hope is that these results, that they provide a first foot in the door for understanding exactly how these combinations of perturbations add up. Okay, so we are now, this has all been published, and we're now trying to more recently generalize these results to also include genetic interactions, to include environmental perturbations. And when we speak of genetic perturbations, this allows us to move surprisingly directly actually from this very basic research questions to really translational challenges specifically in the area of rare diseases, which is a second area that my group is particularly interested in. So just a brief introduction, there are some 6,000-ish rare diseases that have a known, well understood genetic basis, and typically these would be mutations in a single gene. So for a given new patient for which I do not know what the actual genetic lesion is that is causing the disease, the key questions are first, well, which one is the causal gene in this particular patient? Does that help to understand the pathobiological mechanisms and in turn to suggest a treatment option? And from a more practical perspective, which data do I need in order to find this causal gene most effectively? And quite typically, one would collect a wide range of different data sets in a clinical context for a particular patient. How do I manage to actually integrate all these different data sets? Okay, so we can translate these questions into this map-based metaphor that I introduced before, where the basic idea is that we first identify the general network vicinity of a particular phenotype that a particular patient is suffering from, using well-described gene defects, and then search for those variants in the particular patient that are actually close by or fall into this disease cluster. Now, a second, what we're also exploring in this area is, since rare diseases very typically manifest at different levels of organization, so you may have signaling defects within the cell, or you may have very specific cellular phenotypes, let's say in immune-associated diseases, all the way to tissue-specific manifestation. The way we try to integrate it is by using a multiplex network approach, in which we encode all these different relationships of genes in different network layers that really span the entire range from the molecular interactions here at the bottom to phenotypic similarities at the very top. So from a technical perspective, this allows us to integrate data very concretely, but I think it's also, to me, quite interesting from a more basic fundamental perspective to study of how an individual molecular level lesion would actually progress through these different levels of biological organization. Okay, so here's how we actually did it. A PhD student in my group, Pamela, made an incredible effort and test of will to collect a wide range of data sets, to curate them, to find the most appropriate way of mapping this or that data set to extract gene-gene relationships, in finally resulting in 45 of these gene-gene interaction layers and more than 20 million relationships between them. So here's some summary plot of some high-level network characteristics of these networks layers, and we see that they're actually quite diverse. So in terms of the size of these molecular networks, their density, the social biases they contain, which to us at least suggests that they really do contain complementary pieces of information. So now that we have these networks, let's start throwing genes on them. Let's inspect the role of rare disease genes. And specifically, we are mostly interested in their disease neighborhoods. So as introduced before, however, there is an immediate problem though, and this is that for these individual rare diseases, we typically only have 1, 2, or a handful of genes at best, and that is not enough to confidently identify what is actually the precise network neighborhood of this particular rare disease. So how we address this is that we aggregate diseases that have similar phenotype into groups of rare diseases, so that as a group, they really reach sufficient connectivity and sufficient coverage to identify these phenotype neighborhoods. And this cute Voronoi visualization shows the final 26 groups of rare disease phenotypes that we are using, and also how many, what the relative number of genes is. Okay, so now we're finally in the position we have collected the networks and we have collected the disease genes to quantify to which extent these individual groups would aggregate on a particular network layer, that is to quantify how strong their network modularity is. And the results are summarized on this heat map, where dark blue dots would indicate that a particular rare disease or rare disease group is strongly localized on a particular network layer. There is incredibly a lot of information in this plots. I would just like to highlight two take-home messages. The first one is that by aggregating individual rare diseases for which information is typically very scarce into groups of similar phenotype, we can actually recapitulate the same network characteristics as for complex diseases. So most easily this can be seen here, where we find that they, similar to complex diseases that have been studied by network scientists for many years, they really aggregate in phenotype specific neighborhoods, which in turn means that we can now apply the entire arsenal of network-based methods also to rare diseases. And the second point that I want to make is here on the left side, where we have tissue-specific actually networks that we build from tissue-specific expression data, which show that different rare diseases have very distinct patterns of modularity in different tissues. And if you inspect these on a case-by-case more closely, you find that there is a very nice correspondence between known affected tissues in a particular disease and the network modularity that we are quantifying here. So to us, this means that this network modularity can be understood as a measure of relevance of a particular level of biological organization to a particular disease. Okay, so let's see how we can use this. And of course, the first thing that we do is to build disease gene prediction methods, and the almost canonical solution for this is our network propagation algorithms that try to identify broader modules around a given set of seed or around a given seed cluster by sending out random walkers or other diffusive processes. And we can modify these algorithms in a fairly straightforward manner to incorporate the relevance of a particular network layer, so that the random walker would spend more time in a particular layer that is known to be actually more relevant to a particular phenotype. Okay, so to benchmark or to test whether this approach makes any sense in the first place, I think there are two important comparisons to be made. The first one is, what if I would only use the most relevant layer, so the layer that has really the strongest modularity signal? Or on the other hand, why don't I just use all the information available? So why do I try to incorporate this modularity in the first place? And the comparison of this can be found here, where these three different shaded curves correspond to using only the most significant layers, using the most relevant layers, or simply using all layers. And well, we find that that incorporating really this relevance criterion according to network modularity improves the performance, which to us is a proof of concept that this kind of approach has merit in general. So we can now go ahead and really build pipelines that are very specific for the particular application of prioritizing genomic variants in individual rare disease patients, which is summarized here. So we would include a phenotype of a particular patient, both for extracting what are the seed genes that are most relevant, and also for extracting what are the networks that are most relevant. Put this into our, how we call it, informed propagation pipeline, and then rank the individual variants in a patient. And we find that for a cohort of known rare disease patients, we are able to retrieve the causal gene really at the top, or the top 5 ranked positions. The last thing that I would just like to mention is we can also go ahead now and try to use this fact that we have identified a broader network neighborhood to really contextualize the pathobiological mechanisms of a disease and to identify potential new drug targets. But this is a very long story in itself, so I want to keep this for another day, because I want to switch gears one last time and talk about this aspect here, which is how can networks help us in the area of big data biology, or however you want to call it. And the key point that I would like for you to go home with is that networks are extremely useful, not only because they have this arsenal of analytical tools, of computational methods, but also because they have a super intuitive visual representation. And here are some of the networks that I showed you before, and each very clearly immediately conveys a distinct message. So from the centralities here to these disease neighborhoods there, to the distance of these disease neighborhoods, the global shape. So in my mind, these and many, many other examples, they really show that important network characteristics, they can often be recognized by eye, and also the other way around, that patterns that stand out visually often have a meaningful network and biological interpretation. Of course, there are some issues, right? So the first one has been on my mind ever since I was a PhD student, where I came across this network that shows friendships between high school students, and it is visually immediately obvious that kids of the same ethnic background, they stick together, and also that there is another divide between senior and junior high school. Now, I found this in a fantastic review by Mark Newman. If you've never read it, it's a classic, I'd say, where he says, and I quote, "We have no special reason to suppose that this very simple algorithm, a very straightforward spring-based algorithm, would reveal anything particularly useful about the network." So he really does have a point, right? Like, why would I expect to learn something about a network by pretending that the nodes are negatively charged and edges represent springs. So in essence, we really cannot trust what we see, and also the other way around, we can't quite see what we trust. So in the beginning, I showed you this, this map analogy, but actually I drew in all these neighborhoods and these centralities by hand, because if I had highlighted the respective genes in this spring layout, I wouldn't have seen anything, because more generally, if the networks are too large, we get into this hairball hell where we actually don't see anything at all anymore. Now, we would like to propose a solution to these challenges, and that has two components. The first one on the left side is that we're trying to develop frameworks for really having interpretable network layouts. So these would be layouts where you know what a node position means, and also where you can position nodes to reflect any network characteristics that you want, so that you have an immediate connection between, well, the positioning of nodes and their interpretation. And the second part is to use immersive technologies where you can really blow up the size of these very messy hairballs to a cathedral, as my favorite metaphor, in order to really inspect all its parts, going from local structures that you can zoom into to global patterns that you can also observe. Okay, let's start briefly with the first part. I want to kind of rush through it because I also want to show something else to you, which is our framework for making interpretable layouts, and that has 4 steps. So the starting point is we want to do this really for genome scale network sizes. So we don't want to stop with small networks that we can still sort of manually inspect. And the first step is that we convert the network into a feature matrix, so that every entry in this feature matrix describes the relationship between a pair of nodes, and these relationships could be based on anything really. So this could be network distance as obvious candidates, but yeah, also anything else, and I'll give you a few examples of that. Later, we would then use standard dimensionality reduction techniques on these feature matrices, which is appealing because they are very powerful methods available. So it's fast, it's scalable. And finally, we would use different spatial embeddings that will also be useful later on for doing, let's say, 3D printing, or for doing these VR applications that we have in mind. All right, I want to show you, to give you a sense of what these things are, first of all, in some toy networks. So we have here a global layout, which is based really on the distance between nodes. Another layout is based on node centralities, and a third one on the local connectivity patterns. And we see that they produce vastly different layouts, actually, depending on which network on layout modality I choose, and each of which conveys a very different message. So this one here, for instance, shows very nicely very clearly the hierarchy in these Cayley trees. This one I liked because it shows that these grid networks are actually bipartite, which I never realized, and I've been working on grids for 15 years. This one very nicely recapitulates the shape that one would also expect from more spring-based layouts, while being considerably faster. So it's a very versatile and powerful framework. I will skip this because I want to show you something. This is the solution that we have in mind for the second problem, where we really want to go and in an immersive fashion inspect these large-scale networks using VR technologies. And earlier this year, we published a prototype of this. And instead of going through all the specs, I really want to show you how it looks. Typically, I would do this in our green room, but today I'm actually not in Vienna. So I'm going to show you a really brief video where we introduce that I recorded previously.
When I put on this headset, I can see what you're seeing, except that I'm able to look around in it, whereas you see only the angle from a fixed camera position. So here on this side, we have the interactome in the disease layout that I introduced to you previously. So the first thing I want to do is I really want to go inside so that I can look at all the genes and all the clusters. So now these clusters, they correspond to the different disease modules, that is to the local interactome neighborhoods where an individual disease is located. And the first thing I would like to show you is how we use these transitioning features in order to switch between different contexts of genes. So whereas now the disease context is emphasized, I can transition to a functional layout that emphasizes the biological processes that genes and clusters thereof are involved in, and by following up on where individual genes go between disease and functional layout, I can get a...
All right, I have been warned, rightly so, by the chairs of this session that I really should stick to time. And since I made so much blah blah earlier on, I would wrap it up. And you can look at some of these videos on our website. These are some of the things I talked about. And I finally really need to thank the people that really did all the work. Thanks. I apologize for the rush, and I'm happy to answer a few questions before the next talk begins.
Thank you, Jorg. Very nice talk. Thank you very much. I hope in Café Connect we can continue to discuss some of these visualizations. I see some questions coming in already. From Katarina, there's a question. So for the rare disease example, how have you actually integrated such a vast amount of different data? What was the method that was used?
So this was a fantastic effort that took us years, really, because for each of these different data types, they require their own method. So depending on whether you use something that is ontology based, or for something that is expression based, or for something that is direct physical interaction based, we would employ different ways of extracting these networks out of them. I hope that the paper should come out soonish. There is no short answer to that other than we try to be very specific for each different data type.
Thank you very much. I might have one very short question, hopefully, for the perturbome. Did you look at if the drugs that were actually connected were also of similar types, so that the similar types of drug interact, or was there no patterns between the types of drugs? So I don't know.
In general, yes. So we find that drugs, well, of similar type, let's say based on chemical structure, but also based on disease annotation, but also based on molecular mechanisms, they tend to produce similar phenotypes, and they also tend to be closely related on our molecular networks. So yeah, in general, the answer is yes.